Good day, dear students. We are continuing our immunology trip, and today my task will be extremely difficult. Because first of all, we need to continue to discuss basic concepts of immunology, basic terms, basic ideas, and their use in medical practices. We need to discuss main steps, main principles, the scheme of immune response, and how different cells can interact one with each other during that immune response, how to use that knowledge in medical practice again. And we need to talk, we need to explain, to describe all immune methods of diagnosis of infectious disease, how to use, how to evaluate the result, how to perform. And I have only one single lecture for that, so it's going to be a tough story. Please take a pen, take a notebook, take a seat, and take a kilo of patience with you. Let's start. It's not a secret to anybody that the same object in different situations and different descriptions can be provided with different names. So can you tell me please, what is that? What is that thing? I'm pretty sure that if you will show that picture to common people without medical education, they will take a look and say, oh sir, it is a virus. And it's looking like correct. But you already studied microbiology, and you of course should remember that correct name for that is a virion, because virus is extracellular form, virus is intracellular form, and virion is extracellular complete. And that is a virion. Already different name, but let's take a look at different sciences. If you will ask some biology man, what is it? They will take a look, say, it is a microbe, and it is correct because it is microorganism. In geometry, in some maybe school kids, they will say, sir, it is sphere, because it is spherical, and again it is correct. But in physics, it will be given with the name particle. And if you will remember last lecture, previous lecture, we said that in medical practice we will call it as the pathogen of cause, because it is able to cause a disease. But today we need to take a deep look in immunology. And of course our immune system will not care about shape of that thing, or size, or color, or its functions. Because in primitive language, the main task for our immunity is to understand, is that thing foreign or belong to us? And if it is foreign, we need to respond. If it is something our own, then okay and fine, we will not fight with that. And of course, immune system will not recognize that thing as one large. It will recognize it as antigens. And the correct name, the correct term in immunology which is used, it is antigen. So antigen is some substance able to initiate immune response. In immunology, that is the key idea. Looking ahead, many of you will say or will state that any foreign substance. But I'm pretty sure you all know that it is not always so, because of course our own cells have a lot of antigens as well. But our immunity can recognize, understand that that cell is our own, it's fine, it is good cell, and it will not fight with them. But sometimes immunity can be broken, and sometimes that mechanism can work incorrectly, and our own cells or our own tissues can be identified as foreign. Yes, I'm talking about autoimmune disorders. So it's more correct to say just any substance able to induce immune response. Again, immune system will recognize that thing as antigen, but not single, because our immunity works in much more nuanced level. We look at that as a one single common organism. But if you remember our first lectures, we described structure of virion and detailed composition of a virion. And we know that it is made of several different structural compounds. For example, protein couch is named capsid. Capsid is covered with red spikes or peplomers, and it have genetic material, it can have some enzymes, and of course those components are very different one from each other. They similar between identical compounds, but they're very different between each other because they are made of different chemicals, different elements. And of course, if you remember, structure of capsid is made of capsomers. Capsomers are made of different proteins, but structure of every capsomer is identical. Because within capsomer, number of proteins are the same and type of proteins are the same. And of course, they will make one structure. Spikes, glycoproteins. Can be different types, but all are made of glycoproteins. Nucleic acid, DNA or RNA. So for me, when I look at a thing, it is something single because I can see it in microscope as a single spherical particle. I can take that microbe, inoculate in lab animal, and it will cause a disease as one single particle. We can inoculate it in cell culture. It will grow here, it can perform some cytopathic effect because it is one single organism. But for our immunity, it's not so. For immune system, it is a mixture of some independent sets of unique antigens. And our immunity will not fight with that one single thing. It will make unique weapon against every unique compound. And that's amazing thing, I think, because what is it? How do you think? How can you see 1 and 2 different objects. Are they the same or they are different? One is spherical, much smaller in size. Another one is filament or cell elongated, much bigger and looking like different. But actually no, it is one single influenza virus. And it have an option of or a property of pleomorphism because virus can change shape and size. It can vary. And actually both are the same virus. And if you will show that picture for common people and ask, is it one single virus or two different? The answer is obvious. They will say different, of course. They're absolutely different. They're looking even different. But again, our immune system will not care about those properties we are using. It will not take a look on the color. It will not take a look on size, shape, or etc. It will recognize compounds present on the surface of that organ is present inside. And compounds here are the same. Virus is made of the same compounds because it is the same virus, of course. And viruses are pretty complex, and we will study it mainly in next semester. And today we need to start with bacteria. So you all know structure of bacterial cell from our first lectures, and let's name main organs here. What is that? Right thing, tell me please. Or what is maybe that, and that, and that, and that bluish flagella, pili, cell wall. Okay, you are well done. But I believe you all asked yourself in the first class, first lesson, and we really do have that question in exam, chemical composition of a bacteria. So I think you asked, sir, why do we need chemical composition of that thing? Really, we cannot even see it without microscope. Why do we need to study such a petty things? Maybe now you understand, no? Of course, different chemicals will make different antigens. And for us, for me, that one is a cell wall, that one is a flagella. But for our immunity, our immunity will understand chemical composition. Lipopolysaccharide in the cell wall, LPS. Immune system will say O antigen, somatic antigen. Flagella is a set of flagellin proteins, another antigenic structure. H antigen, pili, F antigen, capsule will be identified as a K antigen, and some microorganisms can have again different types of capsules, we said already. In Salmonella, K antigen can be named as VI antigen, virulence antigen. So differences in the structure of certain organelles of the bacterium causes activation of separate immune response. And for every of those organelles, our immunity will produce unique type of antibodies. Again, for us it is one bacteria, but immunity will identify one bacteria as a set of as minimum 4 different antigens, and will induce not a single response but multiple. Moreover, if we will take a deeper look in immunology, the same organelle in different microorganisms can be made of the same chemicals and the same by nature, but with slightly different structure or in kind of different modification. And how to explain LPS, lipopolysaccharide, we again said that endotoxin have the same activity. We combine all types of LPS in one, but lipopolysaccharide, it is a complex macromolecule. It's made of polysaccharide and covalent connected lipids. And how many lipids do you know? Just try to remember. And how many polysaccharides? Poly, multiple. Let's take a look on the structure of monosaccharides. Just take a look here. How many do we have? It's only basic. And structure of every slightly but different. And if you have poly, how many combinations are possible? Of course, a lot. So for us, one LPS, because we don't care about particular structure. But at the beginning of our lecture course, we said that to put a diagnosis and to prescribe a therapy, medical doctor need to know species, of course, species of microbe. That exactly term is used in medical practice. Oops, I'm disappeared, sorry. Species is used in medical practice as the key name of the microorganism. So do you remember how to identify species? Of course, we need to first take a look in that microscope to understand gram-positive, gram-negative, shape, size, and etc. Then inoculate in culture medium. List and study biochemical properties. And by set of 3 types, 3 groups of properties, human can identify bacterium. But our immune cells, they cannot see in microscope. They cannot cultivate, but they can identify different types and different modifications of every microbial antigen. And all antigen can be named as O1, O2, O3, according to which type of modification is it. And combination of all antigens present in a particular microorganism is known as antigenic structure of that particular microorganism. Or we use the term serotype. Serotype is the main unit used in immunology. Species, not really important here, because serotype is more specific than a species, because slight modification in structure of the flagellum or a structure of some cell wall, for example, it will not change appearance of the microbe. We'll still see that as like some gram-positive rod-shaped bacteria. But if we'll put it on culture medium, it will give the same growth, because if flagellum is present, it doesn't matter what components are present here, it still will be some mobile bacteria. And if we will study biochemical reactions, of course it will produce the same enzymes, and enzymatic activity not dependent on structure of the cell wall. So we will not understand that microbes are different, but immunity can, because immunity will not study biochemical properties. Immunity will recognize particular structure. How it is known that serotype is different? We, of course, in nowadays can see the structure of the molecules, literally. We can take a look at an electron microscope and we will see that compounds are different. But serotypes are known for decades. And in those times, we can only dream of such technologies. Of course, no one could study a particular structure of the cell. But we knew that a particular species of bacteria can cause a certain disease. And after recovery, subject will have protection against the disease, immunity, antibodies going forward. If we will give that antibody to another person with the same disease, those antibodies should inactivate pathogen in that ill person, and they will help to recover. Immune therapy, we're using immune serum. Of course, you all again should know about that already. And very often it is really so. Immune serum will help. But some time, at some moment, it turned out that not always serum was working. Why? It appeared that seems like the same bacteria, but inducing not the same immune response. And to inactivate one species of bacteria, you could be needed in different types of antibodies. So to make it more clear to everyone and to give consistency, antigens were provided with numbers. As you can see, every antigen can be given with the number. So the very first type of the cell identified was given with number 1. Next modification described to identified was provided with number 2. And so on, every next modification was given with the consecutive number. And combinations of antigens will define serotype of microorganism. Every antigen with unique number will give you serotype. And I'm pretty sure again you all know some of them. If we'll take a look in bacterial serotypes, we have that E. coli, O104H4, influenza virus H1N1. Everyone knows H1N1, it is Spanish flu. And nowadays more popular name is swine flu. And H1N1, it is serotype, name of antigen and number of their modification. So in the classic version of diagnosis, to identify type of antigenic structure, as you can guess, you need to use serum contain corresponding type of antibody. And in classic definition, serotype is a group of microorganisms within one single species with common antigenic structure. Set of antigens will give serotype. Of course, to identify, to determine serotype, you need to use serologic testing. And serologic testing is a wide group of tests. And we should analyze in detailed manner today, but it is the task of our second part of the lecture, so a little later. If I will rephrase the beginning of the lecture, I said the same object can be given with the different names. For us, world is made of objects. If you will try to understand how we see in the world, you will say that we have some shapes, colors, distance, and if you will take a look into some corridor, you see doors, angles, shapes. But if you will take a look into the Matrix movie, the world is made of symbols. And for our immunity, the world is made of antigens. But here we have two sides of the coin. We took a look at the first one, let's take a look at another one. And another side is much more complex and much more complex organized. It is immunity. Today we need to find out principles of acquired immunity. Last lecture we talked about innate, now acquired. As you remember, acquired means everything obtained during the life, after the birth. And partially we already discussed some features of acquired immunity. Now we need to combine, to arrange our knowledges into one common single system. So I already said system is really complex, and system have multiple actions. It can be provided with 4 major properties. The first property of immunity is a diversity. Diversity is the ability to recognize a vast variety of foreign antigens. And our lymphocytes, they're presented by approximately 30 million different clonotypes. 30 millions, it's an incredible number. Try to remember 30 million symbols. Really, 30 million symbols? I will not remember it for the whole life, but our immunity have that option. So, second property is specificity. Specificity is similar, it's kind of closed term. It is the ability to differentiate between foreign antigens. Specificity is required to avoid inappropriate immune reactions or cross-reactivity, to understand that that and that antigen, for example, are different. And if you have that, don't use that antibody. So it's really, if you will just read definitions, it's confusing because vast variety here, remember, recognize, how to understand. So to make it a little easier, I will extrapolate it on me and you. I have a large group of students, juniors, seniors, Russian, Indian, some other foreign students. And let's pretend I have 1,000 of you, thousands of students from India. And I know every of you. It is diversity. I'm able to remember 1,000 of different students, vast variety. And now your dean calling me, Alexei, can you please come to my office now and take Shubham with you? I have 2 students named Shubham. Diversity? Yes, I have. I know that I have 2 Shubhams, but without specificity, which one should I take? And I have a 50% chance that I will take wrong student with me. I will have inappropriate result, incorrect reactivity. Or I can take 2 students and brought 2, cross-reactivity. Instead of 1, I will punish both. And if I will know which particular Shubham dean is talking about, I will choose that one correct person and bring it with me. That one is specificity. So both terms are required to perform correct functions of immune protection, of course. So third property is discrimination. Discrimination is the ability to recognize and respond to a foreign molecule and to avoid a response on our own body. Speaking simple terms, it is a friend of foe system. In that, if cell is mine, if I understand that cell is belong to my body, my organism, immunity will do nothing. If it is foreign, we need to fight. Looking like simple. And the first property, again, it is obvious. It is a memory. Memory, of course, is the ability to remember structure of antigens and remember how to produce antibodies. So as far as immunity is acquired, our immune system initially is empty. It has no information about any antigens. We deliver it in that world absolutely clear, sterile. So to fill it, we need to introduce some, to give some data. So during the first contact, immunity will study, recognize structure of pathogen, study antigens, generate specific immune response, and eliminate antigen. Infection will be cleared. That one is the first encounter, but it will take some time. Immune system, of course, need time to react. And after the first encounter, immune system retains the memory of that encounter. And in the future, if a second or latest contact will occur, of course, immunity will run much more faster. It will run more quickly and more abundantly than the first episode. So because of the memory, of course. If you'll take a look on possible mechanisms, then you already, of course, know we should have another classification. And according to mechanism, immunity can be cell-mediated or humoral. Humoral means antibody-mediated. Cell-mediated sometimes can be known as a cellular immunity. But of course, very often both comes together, and they depend one on each other. First of all, of course, because antibodies are produced by cells, as you know. Multiple cells have the same origin. And I hope you remember that our blood cells originate from bone marrow, of course. Bone marrow will give parenteral cells, giving rise to other one. And do you remember what are parenteral cells for blood? We have actually two types. It is lymphoid stem cell and myeloid stem cell. So neutrophils, basophils, monocytes, macrophages originate from here. But today we're interested into the first type of cell, lymphoid stem cell. Lymphoid stem cell, if you remember, will give rise to main types of cell. Three we have. First one is a natural killer cell. Natural killers are type of cytotoxic lymphocytes which are critical to the innate immune system. They belong to rapidly expanding family of innate lymphoid cells. Last lecture we described, do you remember? No antigenic sensibilization. Cells are always ready to fight right now during the first encounter, even they're too active. And NK cell is a unique population of lymphocyte. They're closely related to B and T cells, but different. And we already discussed function of natural killers last lecture. So more important today is T and B cell. Initially we have premature B cell, premature T cell. And premature B cell processed in the gut submucosa in mammals. For the first time, that process was studied on birds. And birds have bursa instead of the gut submucosa. It is slightly different organ, unique one. And cells identified in bursa was named as B cell or B lymphocytes. Bursa originated. So B cells, premature B cells, they are still not completely mature. They are ready to fight, but they are waiting for some signal. And that signal is antigen. When antigen will activate immune response, B cells will start to divide. They will produce two clones of daughter B cells. Clone means it is a group of identical descendant cells. This clone, in the case of B cells, is a B plasma cell and a B memory cell. They will be activated against the same antigen. B cells are antigen specific. And that phenomenon when one single cell will give rise to two independent clones, but against one single antigen, is known as a clonal selection. So plasma cell, as you can see, here it is bigger in size, but it is very short-living. It's living only for about few days. And the main task of plasma cell, or we call it effector B cell, it is a production of antibodies, of course, you should know again. Antibodies will be produced in huge quantities. And memory B cell, it's smaller in size, but it is circulating cell. It will remain in bloodstream, and mainly initially it's in inactive state. The main task for memory cell, of course, is to keep information about structure of antigen. Lifespan of that B cell depends on the clonotype, and different clonotypes can have very different duration of life. It can vary from few months to decades, and some clones can even live for lifelong. So memory cells remain dormant until activated by further contacts with the same antigens. Maturations of T cell, as you all know, occurs in the thymus, hence T lymphocytes. T lymphocytes are of two main types: cytotoxic T cell, which are similar to natural killers, but as we already said, that one have antigenic specificity. Cytotoxic killers, they can recognize target foreign cells, fungi or bacteria, for example, or they can recognize our own cells which are infected with the virus or modified by a cancer. And again, you remember last lecture we talked very similar things about natural killers, but those are against particular antigenic structure. So they as well can release perforins on the cell membrane, and they can inject cytotoxic substances directly into the cell, and similar, they will lead to swallowing and burst of the cell. So the most famous type of T cells is, of course, T helper cell. You all know T helpers, and mainly Th cells involved in regulation of the immune response. They can stimulate production of antibodies, they can secrete different multiple growth factors, they can activate maturation of premature cells, and multiple other functions. And according to main effect, two types of T helpers can be distinguished. Maybe you already know. We have Th1 and Th2. Th1 involved in cell-mediated response. They are connected to macrophages and to cytotoxic T cells. They helping to fight with bacteria and protozoa. Th2 cells involved in the humoral response. They help to fight multiple extracellular parasites such as the helminths. In nowadays, we have one more additional type of T cell, T helper cell. It is T17. T17 helpers, given with that number because the main task of them is production of interleukin 17. Again, if you remember that cytokine IL-17, that one is due to that T helper. And mainly they're involved in the protection against fungi. We have actually more number of other T helpers, and they are already described. But it's not, again, the task of our department to give you such detailed classification. So T regulators or regulatory T cells formerly were known as T suppressors. Original name was given because the main function of that type of cell is inhibition of previously activated immune response to immune cells, cytotoxic T cells and T helpers, to prevent exhaustion of our immunity, to prevent damage of our own cells, and to stop hyperactivation of immune response. Again, we talk about cytokine system that if you activate it, you need to stop. And the task is for T suppressors or T regulators. So recently it was shown that T regulators can manage activity of different types of body cell. And therefore, current title was given T suppressors is still in use in some literature, but mainly in outdated literature. And nowadays, more correct name is T regulator. We have as well memory T cells, and they're similar to B cells. They are formed in the response of the contact with antigens. They remain dormant in lymphatic tissue, and they keep in the memory about structure of antigen as well. They can be found in lymph nodes and spleen, and lifespan is for years. In the case of the second and furthest encounters, memory T cell will proliferate. It will start to multiply, and it will differentiate into T cytotoxic cell, T helpers, T regulators, and can give rise to additional T memory cells. And of course, it will enhance duration of enhanced speed of immune response. B cells will start, B memory cells will give rise to B plasma cells and secrete antibodies. T cells will activate cell-mediated immune response. So Now you are familiar with the main participants of immune response, and we need to understand way of their communication, way of their progress. So assuredly you know the main process of immune protection, phagocytosis. You all know phagocytosis, originated from Greek words phagein and kaitos. We can translate it, or it stands for eating cell. And initially, name was given by Russian scientist Ilya Mechnikov, again 1882, to reflect that ability of a cell to engulf, to eat foreign particles. And it is our human cells. It's looking like amoeba, some protozoa, but human cell. And of course, if you will ask any human, what type of phagocytes do you know? And of course, many number of people will say macrophage, because it is largest cell which is present in any human tissue. And of course, it can perform multiple functions. Phagocytosis is only one of them. And process of phagocytosis occurs in several stages. Maybe you already know, but we need again to refresh our knowledge. So first of all, you have macrophage, you have that foreign particle here, near macrophage. And presence of that foreign particle near macrophage will initiate chemotaxis. Chemotaxis means cell will start to move and get very close to foreign particle. Second stage, adherence of the particle to the surface of the phagocyte. So to engulf, of course, phagocyte need first to catch that microorganism. So second stage is ingestion. Sorry, first stage is chemotaxis and adhesion. Second stage is ingestion of the particle. So of course, that foreign organelle, foreign particle will be engulfed in that vacuole. And you will have formation of phagosome. Phagosome is presence of that presence of that foreign particle in a lysosome within the macrophages. So antigen is engulfed and trapped within the cell. But to destroy that thing, of course, macrophage need lytic enzymes. And those enzymes are so active that in free state, if they will just swim in the cytoplasm, they will destroy the host cell. And macrophage, of course, do not want to die. And to remain alive, those enzymes are trapped in a specific vacuole as well. And that vacuole is named as a lysosome. Lysosomes are small. Vacuoles contain a lot of enzymes. So phagosome will migrate towards each other and merge with lysosomes. And they will form large one big vacuole, which is named phagolysosome. In the fifth stage, digestion will occur. And after digestion, sixth stage, formation of residual body will act as a name stage. So remain indigestible debris will be formed, and of course cell need to reduce and to remove them. In the seventh stage, all waste materials will be released outside of the cell in the environment. So that process of phagocytosis is well known for you. You routinely studied in, I think, in biology maybe course, but what is not always provided, what you cannot know. First of all, as you know, every cell is made of chemicals. Every chemical has a charge, positive charge or negative charge, right? So cell, one cell, every cell, macrophage, foreign cell, in general, have a charge as well. It is a summary of every chemical cell charge. So initially, if we will take a look at macrophage and bacteria, they have negative charge, both. Why is it the problem? Of course, you know that if two particles have identical charge, they will disattract, and it will be very difficult for our macrophage to move to a pathogen because they will repel each other. And those pathogens can avoid catching by macrophage, avoid destruction, and it will continue to replicate inside our body because macrophages can run on continuously. So to make phagocytosis easier, to help macrophages not to waste so many powers, our immunity can perform opsonization. We already talked about opsonization slightly. It is an immune process in which opsonins are used to tag foreign pathogens for elimination by phagocytes. So when opsonins will cover bacteria, you will have something like that. So opsonins are substances that can bind to a pathogen, and they have a receptor which can identify and bind to immune cell. So when pathogen is covered with opsonin, phagocytes are attracted to the pathogen. And of course, our macrophage will be much easier to take that bacteria and perform phagocytosis, of course. So if you'll ask what examples of opsonins do we have, complement. Of course, we have complement as the main system to perform opsonization. Some immunoglobulins can perform as well. And if you remember the diagram I showed you when I showed you the scheme of classic way of complement activation, that C3b proteins of complement system can perform opsonization. They involved in process of enhancement of helping to phagocytosis. Again, complement system because they complementing phagocytes, for example, to perform phagocytosis. So next thing about phagocytosis we need to concentrate our attention on. We mentioned before that macrophage have several functions, and besides phagocytic, it can perform function of antigen presentation. And very often macrophages are named as APC, antigen presenting cell. So when we describe that process of phagocytosis, I outlined only a part of the process, because if you will take a look on that stage when phagolysosome is formed, it's not actually completely so. And routinely that process is not mentioned because it's not really important to understand the principle of destruction. But if you will take a detailed view, microorganisms or some foreign particles, of course, will not be destroyed immediately, because how to show it here, if you will see that blue lysosome with the yellow particles, lowest one, initially one large object is broken into the smaller identical compounds, and every small unit, yellow unit, is of course antigen. And macrophage knows that, and it knows that that thing is trying to invade. And maybe that object is not single here, maybe you have a lot more around. And of course, one macrophage will not be sufficient to stop that invasion, and the whole immune system should be aware. And to aware immune system, we need to show antigen which is trying to invade. That antigen should be presented to cells of our immune system to induce specific immune response. So to introduce that antigen, macrophage has specific vesicles with the molecule of major histocompatibility complex, or MHC. You again all know them. So MHC is a large group of surface proteins present in every cell of our tissues. They are needed to differentiate between our cells and foreign, that way histocompatibility. And originally MHC was discovered when doctors started transplanted tissue compatibility. So, of course, macrophage is our own cell, is completely whole, covered with MHC molecules as well. And other cells can recognize that that one is ours. Okay. And it is kind of identification mark to surrounding cells. So, cell will study MHC, and I'm here, I'm local, don't fight with me. But of course, macrophage now need to present foreign antigen. And antigen molecules should be fixed to MHC molecules. And in that lysosome, you will have another additional step. Antigens will form a complex with the MHC, and those molecules will be presented on the surface of the cell. As you can see, antigen is presented here, some here and here. And now then, primary immune response will be studied. Presentation of antigen by macrophage will initiate immune response. So I will try to show you that schematic. Again, view antigen was identified by macrophage. It will be put at some here with the help of MHC. Presence of that foreign molecule on the surface of the macrophage will initially attract naive T cells. We said already. And of course, T cells will start their activation. CD8 T cells will be activated to clone and will give rise to cytotoxic cells and memory CD8 T cells. Cytotoxic cell will help macrophage to destroy antigens initially. And antigens, again, some here, will be engulfed by cytotoxic T cells. Sorry, where are you? Hey, not working. Here. So CD8 cells will as well give rise and form memory cells and T helper cells. Of course, activation of CD8 cell will give a signal to release multiple cytokines. Those cytokines can attract more, how to stand, you will see it, will attract more macrophages and natural killers to the site of invasion. And of course, all set of cells will fight with initial antigen, directly with antigens. Such-wise, more macrophages will be involved in the protection. They will activate more naive cells, and they will attract more other cells. And that's how immune response is going in the cascade. So the main task is, again, to fight with antigens. So that type of immune response is known as a cell-mediated, because a lot of cells are involved to destroy directly those antigens, killer cells, cytotoxic cells, macrophages, and etc. But of course, we have another option. And of course, B cells should be activated as well, as you know. And immature B cells are waiting for the signal. And when they will be activated to clonal selection, B cells will give rise, of course, to plasma cells or effector cells and memory cells. And memory cell, as you know, will keep information, but now we need that plasma cell. The main task is secretion, production of antibodies, and they will be secreted in big amount. And of course, they will help to perform opsonization and to inactivate pathogens fast, and that one is humoral immunity. So as you guess already, the main unit of humoral immunity is antibody, of course. By nature, antibody is a blood protein of globulin group. It is produced in the response to an encounter to a specific antigens. And that one is a schematic view. Usually it is a Y-shaped, and in common, again, literature or common some slides, antibody is shown as some organ, something matching by structure antigen. And antibodies are secreted form of the B cell receptor. And initially, antibody is identical to the B cell receptor that secretes it. So We have that B plasma cell which is covered with antibodies, and when antibodies need to be secreted, they just detached and get into free swimming. And the identical two receptors remain on the surface of that B cell. And as far as antibodies are globulin produced by immune cells, very often, of course, they're named as immunoglobulins, or abbreviation is IG. And that is the classic schematic U, Y-shaped molecule. And it is shown like that because it is real shape of the molecule. And if you will take a look at schematic U, it will be more correct to say to show it something like that. So structure of antibody is symmetric, as you can see, and consists of two identical heavy chains marked with the green lines and two identical light chains marked as red lines. So it was shown that different antibodies can have different structure of chains, and it was shown that two types of heavy chains are two types of light chains are possible, lambda and kappa, and 5 types of heavy chains, alpha, delta, gamma, epsilon, and mu. Why again it is important? A little later. So all chains are connected together with disulfuric bonds, and it is required to make structure of antibody flexible. That region is known as a hinge region. Two more regions identified in the structure of antibody. It is FAB, or fragment antigen binding region, and lowest one is a fragment crystallizable region, or FC region. So obviously the first is required to fix antigen, and as you can see, antibody have two FAB regions. So one molecule of antibody can fix two molecules of antigens. It is important, please remember. And as far as different antigens can have, of course, different size, they can vary in size. Some can be bigger, some smaller, but size of antibody is fixed. Of course, that flexible region is important to have, because antibody can kind of adapt to the size of the antigen. If antigen is big, it can become wider and fix still two molecules of antigen. So fragment crystallizable region, or FC region, is required to bind phagocyte or complement proteins. So do you remember that classic way of complement activation, when again I showed you antibody antigen here? They will form a complex, but to bind they need that C1 portion of complement system, C1 protein. Then C2 and C4 cleaved to A and B fractions. Yes. Okay, you should have a good memory to become a good doctor. Please remember that. So let's continue. We said that antibody, it is a receptor of a B cell, which is required to bind to a phagocytic cell. And our cells have pretty constant structure. So if it is secreted to the contact with antigens, and antigens can have different size and different structure, that one is stable. We have common, that idea is displayed in the structure of antibody as well. So we have variable region, that yellow showed with the yellow mark, which depends on the structure of antigen, and we have a stable region, that large one. It is constant, means that structure of any antibody is the same. If you take multiple antibodies against different antigens, constant, the same. Structure of that corresponds to the structure of antigen. And as you can guess, of course, that variable region provides specificity to our immunity, to our antibody. As you know, of course, we have 5 classes of antibodies. Tell it, please. M, G, E, A, and D. Okay. And routinely we name them as IgM, IgG, and so on. But do you know what is the difference? How different antibodies are different? No? So let's take a look. As you can see, come on, I will hide. IgG is really white shape. IgE is slightly bigger but still white shape. And IgA and IgM are not white shape, but here, and the last one, IgA is a dimer made of two identical Y-shaped subunits, and IgM is a pentamer made of 5 Y-shaped subunits. How is it important? Take a look. IgA has not 2 antigen binding sites but 4. And IgM, yes, 10. Molecule of IgM can fix 10 antigens. That antibody is 5 times more active than common Y-shaped structure. And if you clap your eyes on the chart, you notice that type of heavy chain conform to the class of the antibody. Yes, alphabet is different, Greek and English, but you see G class, where is G? Here, G class is made of gamma chains, E, epsilon, M, mu chains, and of course pentamer have the heaviest mass, it is the biggest one, and because very complex structure, it's shorter in life duration, and monomers are of course lighter and they are more stable. They are the main types of antibodies found in blood of humans, and IgG is prevalent, as you can see from the chart. So IgG is the most stable. It can keep activity for months, even for maybe years. And because it have the smaller size, even here I'm trying to show schematically proportion of different antibodies. IgG can pass through placenta, and IgG can provide, it can give protection to the fetus, if you already know. And up to half of the year age, fetus are protected by mother's IgG. IgE, as you know, is involved in the early G, and it is because Fc region, if you will take a look on the chart, of E class is mentioned receptors of the mast cell and basophils, but not phagocytes. Here it is shown. I will take it away. So IgA can be of 2 types. One is circulating in our blood, providing internal protection, and another one is secreted in the body fluids and provide surface protection. So very often it is marked as IgA to differentiate easier. S means secretory immunoglobulin A. So the most observant of you noticed that main function of IgM, here it is primary immune response, and IgG secondary immune response. Inquisitive students already ask themselves, sir, what is it? What's the difference? What is primary, what is secondary? Immune response is still immune response. So let's find out. In current slide, I draw some kind of a scale. You can see days, you can see antibody titer or amount of antibody. Let's pretend that it is some life period of some person. Let it be some completely random person, no one knows, namely me. Today, I'm standing here in a 0 day. Let's take some disease, any disease I never had a contact with, neither ill nor vaccinated. And in the view of the fact that my immunity is not familiar with structure of that antigen or with the antigenic structure of the pathogen, which is trying to cause a disease in me, of course amount of my antibodies is equal to 0. And it is shown in the diagram, nothing I have. So today, I had a contact with my friend, hello, and I met the pathogen. For me, that contact will be the first one. And of course, my immunity will need to respond first exposure. But as we said, first exposure needs some time. Cells will be activated, but they need help. And antibodies is the good help. So of course, I need some antibodies. And it is already shown here, but how do you think? Why IgM will appear as the first? Why that type of antibody I need immediately after the contact with some pathogen? So, to prevent an invasion of our body by a pathogen, of course, pathogen need to present in large quantity. And it will have some incubation period, and you know that the time is required to accumulate. And of course, our immunity at the very first way will not be ready to respond in specific way, because it have no information about structure of pathogen. So, of course, it is a very high chance that the pathogen will invade fast, that it is present in high amount, and we need antibody as soon as possible with the highest possible activity. We need a lot molecules to inactivate large number of antigen, and of course it is IgM. Yes, it has shorter duration of lifespan, but now the task is to save the response. We need to do it now. We need to inactivate as many antigens as is possible right now. We don't care what will happen in the future, because now microbe is trying to invade. And of course, IgM have 10 antigen binding sites, remember? And that class of antibodies will appear first. Infection, contact, IgM. Of course, activated B cells will produce more and more and more antibodies, but we said that they are very short living cells, and that will occur maybe for the first week. Amount of antibodies will rise, as it is shown here. But of course, numeral superiority of the pathogen and speed of invasion will decrease with the time. Some amount will be inactivated, and immune response will be suppressed by T regulators. So I didn't show it on this scheme, but I do believe you'll remember that our immune system is self-regulated. I repeated for several times, I know. But it is important to inhibit immune response, not to allow to overgrow. And when amount of immune cells and antibodies will be sufficient to prevent progress of the disease, activation of new cells will be suppressed. It will be stopped by T regulators. And a few slides before, I mentioned that plasma cell is a short-living cell, but very active. And of course, they will secrete a lot of antibodies, as is shown here, but after they will die, amount of IgM will start to decrease, as is shown here. So new antibodies are not produced, and those who are already secreted, they will be wasted on the fight with the pathogen, or they're too complex in structure, and some will break down by natural reasons. Complex protein molecule cannot survive for a long time period. It will be reutilized. But of course, you all understand, and we understand, and our immunities understand, that it is still a chance that some amount of pathogen can left on the surface of the tissues, or maybe it's somewhere around us. And of course, microbe can try to invade again very shortly, or maybe some microbes hide it in our body and remains unidentified, but they're already within the body. And I can't just completely remove protection. I cannot remove any amount of antibodies. I need some antibodies, maybe not so active, maybe not so fast fighting with the pathogen, but they should be long-living. And what class of antibody is very long-living and active and can keep memory for a long time period? Of course, IgG. And almost the same time when production of IgM, new IgM will stop, IgG will start to appear, and they will be produced for a few more days. After suppression of immune response, overall immune response, IgG will start to be secreted by plasma cells. And of course, they will help to clean remain small, but some remain infection. Some IgG will be used, and that's why again you will have that decrease in amount of IgG. But some threshold level will remain even after the recovery. And that state, I showed approximately a month, but that is the primary immune response. That stage is known as a primary. So let's pretend a few days later, I got well, I'm feeling fine now, and I went on the street, in the park, somewhere in the shop, in the Maxi, and I met the same person again. Or maybe I bought the same food I'm eating, and now I have second exposure to the same pathogen. What will happen now? What class of antibodies I need now because I met a pathogen again? IgM, of course. I need the same class of antibodies, but now. And of course, I hope that it will appear faster. I will need some time, few days maybe, to activate B cells, but now you can see two important differences. Even here it is shown. First, that period between exposure and appearance of antibodies is much shorter. And of course, my immune cells are already familiar with the structure of antigen. They do not need time to recognize, to understand the structure, to present. They don't need to familiarize themselves with the structure of antigens. It will appear, of course, faster. So second difference, as you can see, I have that amount here and that here. Much bigger in amount, much bigger in titer. So how do you think, what will happen with IgG? Almost the same trend, yes, but even better. IgG will start to rise together with IgM first. Time period is the same, and of course you can see that it's almost higher than me. Amount of IgG will be much greater during the disease, and even after the disease it will be much greater. And that stage is known as the secondary immune response. So you can see obvious differences: time, amount, threshold level. What? I can hear your favorite question again. Why do you need to study that diagram? Why do you need to know when, what antibody appear, how long? Let's shift back to the previous slide, to the main task of the medical doctor, if you remember. Tell me please, what are main tasks of the medical doctor? Again, I always say that for me, we have three, and you should have three main tasks during your medical practice: diagnose in timely manner, treat now, and prevent in the future. And of course, we are talking mainly about infectious diseases. And as we know, classic way of diagnosis of infectious diseases is identification of causative agent. And it is a good method to find out microorganism, identify species, but not always it can be so. What do I mean? For example, what if you have some difficult for cultivation microorganism, or some pathogen which is very slowly growing, or maybe it is some rare pathogen, unique, extremely rare pathogen, and you don't have required culture medium, you don't have diagnostic tests to identify. What? PCR? Yes, it is your favorite method. And method is really good. And nowadays that method is widely used. We can use it, that method as well. It is for sure. But we know that in some diseases, in some cases, pathogen cannot be detected by PCR, or it can be missed in some known stages of the disease. And if in previous years it was kind of difficult to explain to you, but now because of that COVID-19 pandemic, we have the perfect example. A lot of PCR negative tests in ill person. Or if it is an infection of some internal organ, CNS for example, you have no proper education to take CSF. Or maybe state of the subject is so severe it not allows you to take CSF. Or maybe you don't have any qualified doctor nearby you and you cannot obtain CSF on some internal organ structure. What will you do? What are you going to do then? Of course, if you'll take a look on that, it is always an option to take a sample of blood. Any doctor can do punction and venipunction, of course. It's easy, even nurse can do that. And of course, if you will find presence of targeted antibody, you will have your diagnosis. Moreover, if you will take a look on amount and ratio of IgG and IgM, you will understand that you can identify is it a primary response or secondary. What stage of disease is it? If you have no IgG but some IgM, it is first encounter and it is only the beginning of the disease. If you have less IgM, more IgG, it is maybe stage of recovery. If you have that amount, it is for sure not the first episode. So, treatment. It is not the task of our department, but we will skip it. But looking ahead, prevention. It is the topic of future lecture, but now you all already, I'm sure you all know what is vaccine. And without going into details, the main task of the vaccine is to provide information about structure of antigens to our immunity. And immunity should keep that memory about antigens in the future. So looking simply, but now we know that memory is kept by mainly B cells. Antibody protection is kept by B cells, and cell-mediated memory is kept by T cells. And of course, you know that every antigen will initiate new individual response. And new process of clonal selection will be initiated every time for B cells and for T cells. And the problem is in the details. Different clones of lymphocytes, as we said, will have different duration of lifespan. Not only type of information and quality of information is different. They can vary in duration of life. And of course, some will be lost in months, and some will circulate for decades in blood. So, why is it important? Maybe you sometimes wondered why in some diseases immunity is life-lasting, but in some, in measles for example, or if you know that hepatitis A, one infection, lifelong immunity. And in other, influenza or the same COVID for example, it's about for a year, maybe even less. And if you're familiar with that national schedule of vaccination, why in some cases single dose of the vaccine is required, but in some cases we need a booster dose or several booster doses or few booster dose and revaccination later. Why? Now you can understand that it is because of the clonotype of our lymphocytes. Different clonotypes can have different duration of life. If clonotype will be lost, immunity is lost. And to know how strong immunity is, how long immunity is, and how to perform correct scheme of vaccination, how many doses, how large is the interval, when to give revaccination or booster dose. Of course, we need to evaluate quality of the immune response. And we can measure amount of antibodies to understand if immunity is present here or we lose it already. Do we need to give a vaccine, or it is enough amount in our body and we can delay it and give it a little later?